Hi dear students, welcome back. This is Dr Hadi here and welcome back to my channel Medical Globe by Dr Hadi. As you know, in this channel we discuss the basic pharmacology, and now we are going to start the biochemistry lectures. Of course, a lot of topics have been already delivered from the biochemistry in English. Now I am again going to start biochemistry session. So in this lecture and in the upcoming lecture, hopefully we will focus on biochemistry lectures. So today's chapter is enzyme. 2 or 3 lectures have already been delivered. This is the 4th lecture from the enzyme, and that is very simple lecture. It is the history of enzyme, and some students, I would like to request them to please focus and concentrate, because they usually demand the language, Urdu language, which is very difficult for me in this channel. For Urdu lectures, I have already my another channel, but for English, this channel is available. So biochemistry will be delivered in English. So please concentrate. Okay, the history of enzyme. This is today's topic. Very important. Why we study the history of something? Because every scientific knowledge that is now at its peak, once it was very simple. Very little knowledge was available about that thing. So we will read the history, that how this simple knowledge convert into a big knowledge. Like enzyme is an interesting topic nowadays. Because of enzyme, the biochemistry subject has been developed. Because of enzyme, a lot of drugs have been delivered into the market because of enzyme. So the history of enzyme is very important. In simple words, we must know the history of enzyme. Okay, in that history you will get some name of scientists and some basic concepts. The very first thing is here, the word biological catalysis. Biological catalysis. A catalysis is a term that is used for the breakdown. Chizun ki todpod. When things are broken down, we call it catalysis. It's a simple word. And that word came into being in the late 1700th century. Now you know it is 21st, right? 2021. But the first word, biological catalysis, came in that 1700. Means at that era there was no concept of enzyme. Nothing was there. No concept of enzyme, cofactor, reaction, nothing was there. So the first that word came. And how that word came? When they were studying in the stomach, let's suppose we say this is your stomach, right? This is your stomach, and in stomach you people eat meat. Here is meat, and that meat is broken down into smaller particles. A little bit meat is digested in your stomach. So this is a sort of catalysis, because breaking process has been observed here. So the word catalysis first came into 1700 by observing the digestion of meat in the stomach. So this is the first thing. Then in 1800th century, this was in the late 1700. Then in 1800th century, means you can say after 50 or 70 years later on. And one thing more, that scientific knowledge grow very slowly in the beginning, very slowly. Look, there is a difference of more than 40 to 50 years. We learn just one idea in years, but once the science reached its peak, then the speed, it becomes so overspeeded. We get knowledge nowadays, you know, we get knowledge so fast from different sources. So this was the another idea was that starch is what? Starch is a kind of carbohydrate. Starch is a kind of carbohydrate, and that starch also undergo catalysis, means breakdown. Where? In the saliva. So starch is also broken down, breaking process by the help of what? Saliva. This was another observation. Now nowadays you know that this is very simple to say. Oh my God, yes, okay, starch is digested in the saliva. So fine. But this idea was a very special idea at that time in 1800th century. Okay, and then this scientist come, the most famous scientist, Louis Pasteur. This scientist came, and he gave idea of what? Idea of vitalism. What is how people make alcohol? Means sharab. How they make alcohol? Alcohol is made from the sugar. Means some process is given to the sugar, and as a result of this process, the sugar is converted into alcohol. This is called as fermentation. And the Louis Pasteur recognized that the sugar and alcohol conversion was by the help of yeast. By the help of what? By the help of yeast. And yeast is what? Yeast is a cell. Yeast is an organism, a living organism, is a cell. So Louis Pasteur said that the process of fermentation, means the making of alcohol from sugar, the making of alcohol from sugar is because of something inside the cell, because of something. There is something inside the cell. Who said? Louis Pasteur said there is something inside that cell, and that something, he called it ferment. He called it what? Ferment. He said that because of this ferment, the sugar is converted into alcohol. What he said? Because of the ferment, sugar is converted into alcohol. Okay, this is fine. This idea still persists nowadays. But he said this ferment is the essential part of the cell. You cannot separate it. You cannot separate the ferment from the cell. This ferment is the integral part of the cell. You cannot separate, inseparable. So this idea was called vitalism. Vitalism means if you want to convert sugar into alcohol, you need yeast cell. Without yeast cell, without this living cell, you cannot make alcohol from sugar. In other words, you cannot separate ferment from the cell. So this idea was called vitalism. Okay, this idea is going to be rejected in the future now. How? In 1897, look, 1850 and 1897, another scientist came called Edward Buchner. He rejected the vitalism theory. How? He took yeast cell. Yeast is a cell. Yeast is a cell. And what kind of cell it is? It is a fungi. So from that yeast he obtained extract. Extract is just like a juice. It's just like a juice. You take some leaves, right, herbs, right, and you crush it in a beaker or some sort of a mortar and pestle, and then you get the juice. In simple words, it's juice of something. That juice is called the extract. So now you have the yeast cells, but you get the juice from the yeast. The yeast cells are now separated. Now this juice, that extract, we call it extract. With the help of this extract, we convert sugar into alcohol. Sugar into alcohol. So this is called fermentation. Again, this is called fermentation. Alcohol is now ready from the sugar, but this time not from the yeast cell, not from the yeast cell, but from the extract. This extract is obtained from the yeast cell. So the most important thing which is responsible for the conversion of sugar into alcohol is of course present in the extract, is of course present in the extract. You can also say that those things, those some things which were present in the cell is now present in the extract. It means that the ferment is present in the extract and we have separated the ferment from the cell. So the idea of vitalism has been rejected because he said that you cannot separate these from the cell, but now we have separated the ferment from the cell in the form of extract and we got the juice, and that sugar was converted into alcohol. So the vitalism theory has been rejected. Then the extract contain molecules. Now this extract contains something which you can call ferment, and you can also say that the extract contain molecules. Now you will not say something. That something is now called the molecules, and you know very well what are molecules. They contain molecules. These molecules which are present in the extract, they were first called as enzyme. First they were called as an enzyme by a scientist called Friedrich W. Kohne. Friedrich W. Kohne is the name of the scientist, the first scientist who gave the word enzyme. Enzyme is a word, enzyme is a name that first given by the scientist. After this whole drama, that scientist called the word enzyme. Okay, fine. Now another scientist came, his name is James Sumner, 1926. Now we are in 1926th century. What he did, he isolated, means he just separated the enzyme. Okay, fine, he isolated the enzyme. First, he separated the enzyme. And also what he did, he crystallized. Many students, they don't know what is crystallize. A crystallize is a word used for a solid substance, that anything that has proper shape, three-dimensional shape is called crystal. So he also got the enzyme in the form of crystals, like sugar that you use, right? The common table sugar, chini, that is called that is a sort of crystal. The salt, namak, NaCl, sodium chloride, right? You use NaCl during breakfast. It is also crystal. So a lot of solids are available in the form of crystal. So enzyme was obtained in the form of crystal by this scientist James Sumner. Okay, so what is this shape? This circle represent enzyme. This represent enzyme, and in this state it does not have proper shape, so it is not a crystal. Later on, this enzyme was converted into proper shape. I just, for the sake of convenience, yes, for the sake of simplicity, I just draw this diagram, and this diagram shows what? Crystals, right? Crystal. So enzyme is obtained in the form of crystal. Now once enzyme is converted into crystal, a lot of studies can be obtained from it. Lot of studies. In the beginning, only 1 or 2 scientists were interested in the study of the enzyme. Slowly, many scientists started taking interest in the study of enzyme. As a result, the area of the enzyme became broadened and broadened and broadened and broadened. Now enzyme has a vast area, a lot of area, because of many scientists came into that area and they start digging out more and more about enzyme, because we are getting something very interested slowly and gradually with time. He postulated that all enzymes are made of protein. Now this scientist, he has done three things: separate enzyme, isolate enzyme from the source. Enzyme may be present in some kind of fruit, in some kind of a seed, in some kind of leaves. So what he did, he just separated these from, and he obtained crystal. And third, he said a statement, he delivered a statement that all enzymes are made of protein. All enzymes are made of protein. This was a statement that in the beginning, that statement get a lot of criticized from other scientists. Other scientists were thinking, what are you saying, man? How is it possible that all enzymes are made of protein? Are you a magician? क्या तुम एक जादूगर हो जो ये कहते हो कि तमाम के तमाम एंजाइम प्रोटीन के बने होते? क्या बोल रहे हो? तो आपने सिर्फ एक एंजाइम को स्टडी किया. You have studied only one enzyme, and on the basis of this one enzyme, you are saying that all enzymes are made of protein. Are you what do you say? Are you a nonsense? So, some criticism has been hit on that person. Now, how that criticism has been removed? Another scientist, John Northrop and Quetta, these are another scientists, two scientists, right? 1930. They crystallized pepsin. Pepsin is an enzyme inside your stomach, and they also crystallized trypsin, and they also obtained other enzymes which are present in our stomach and intestine, which are involved in the process of digestion. A lot of enzymes are involved in the process of digestion in our body. So these two scientists, they separate all these enzymes and they look into all these enzymes. They realize that all these enzymes are made of protein. So after their work, finally they had to accept that yes, Mr. James Sumner was saying right. He was right. He was saying that all enzymes are made of protein. So finally an idea came. That okay, all enzymes are made of protein. So this is just a statement that nowadays the student of class 9th, he is well aware of this statement, that yes, all enzymes are made of protein. So this statement came for the first time to us in 1930. Fine. Then another scientist, J.B.S. Hall, then. He's another scientist. Once a lot of research has been done on the enzyme, finally another scientist came. He wrote a book where he discussed a lot of information systematically in a proper way on the enzyme. Means he has given lot of statements to prove the existence of enzyme, to prove that yes, enzymes are made of protein, to prove that yes, enzymes are involved in biological catalysis. So a book was prepared, and the title of this book was Enzyme. So the book is called Treatise. The book name is called Treatise. Any kind of that book is a kind of research book, right? So that was J.B.S. Halden, that he collected the ideas of the scientists, all the past scientists, their ideas. He collected these ideas and put into a single book and name it the enzyme. Nowadays a lot of books are available on the enzyme. That scientist also said one thing, that there is a weak interaction between the enzyme, like this is enzyme, and the substrate. A substrate is anything on which the enzyme acts, right? So he said that some sort of weak interaction must be there between the enzyme and the substrate. That statement was very weak at that time, because many scientists they criticized that how you say that substrate enzymes have weak interaction. So no strong proof, no strong evidence was available at that time to prove that yes, enzyme and substrate has some sort of interaction. But later on with many experiments, this become clear that yes, enzyme and substrate has some sort of interaction between them. So that was J.B.S. Halden. And then in the 20th century, research on enzyme has been intensive. Lot of now the overall effort of these scientists gave us a lot of knowledge later on. As I said earlier, that a lot of scientists came into that area because this area was obtaining more and more focus. Lot of scientists were taking interest in that area. They desired to work on that area because this was a very attractive area to study, to work. So the history of biochemistry, the history or the scope of biochemistry is related with the history of the enzyme. With the passage of time, a lot of knowledge was obtained from the enzyme. Similarly, with the advancement in the enzyme, the subject biochemistry get a lot of interest. So the history of enzyme is related with the history of the biochemistry. If sometime you write something on the history of biochemistry, you can put 50 to 60% history of the enzyme, because the history of enzyme has broadened the area of the biochemistry. Now, finally, this was the history, but still something is available to us. Most enzymes are protein. Why now I use the word most enzymes are protein? Because with the passage of time, we came to know that there are very little amount of enzyme that are not protein, that are actually RNA. So except that few enzymes which are made of RNA, all other enzymes are made of protein. So that's why we say most of the enzymes are made of protein. And majority of the enzyme, let's suppose if I say this is one enzyme, majority of the enzyme, they perform catalysis. They perform catalysis, means they just break down things. Catalysis. Catalysis. But there are some enzymes. There are some enzymes, right? There are some enzymes which cannot perform catalysis by themselves when they are alone. They need some kind of friend. They need some kind of partners. And this partner, if I attach here a partner, that partner can never be a protein. This partner is always a non-protein part. This non-protein part, which is the friend of the enzyme, which is very, very necessary for the enzyme. Without this partner, enzyme cannot work. So this partner, this non-protein part is called cofactor. This non-protein part is called cofactor. I think you got the idea of what is cofactor. Not all enzymes need cofactor. Some enzymes need cofactor. And so cofactor, the non-protein part, may be inorganic or organic. In case of inorganic, it may be some metal ion, it may be let's suppose it may be magnesium ion, it may be nickel, right? It may be cobalt, it may be a metal or non-metal. It may, majority of the inorganic cofactors are positive ions, but some negative ions can also be the inorganic cofactors. The organic cofactor may be tightly attached to the enzyme or loosely attached to the enzyme. The one which is tightly attached, they are called as prosthetic group. Prosthetic group. So the definition is very simple. If someone asks you what is prosthetic group, so your definition will be, your answer will be, the cofactor which is tightly attached to the enzyme is called prosthetic group, and the cofactor which is loosely attached to the enzyme is called coenzyme. Okay. Most of the organic cofactors are obtained from the vitamins. In the upcoming lectures, we will study vitamins as well, because vitamin is also the part of our syllabus. So once we finish this chapter of enzyme, we will come to vitamins and we will start vitamins. In a nutshell, Today we discussed the summary, the history of enzyme. We discussed how the word biological catalyst came, at which time for the first time the word catalyst came, then how many scientists they worked, what ideas were generated in the beginning, what ideas were rejected, and what ideas were accepted later on. The name of the scientist and date has been mentioned on the whiteboard. I can just move myself a little bit so that you can have a screenshot from your mobile, or you can just pause the video and start writing my whole lecture. This was the lecture, history of enzyme. I hope you like that lecture, you got that lecture. You give us a like for the courage so that we could record more lectures for you regarding the enzyme in that simple language, English language. Thank you. See you again.